got interphase, which is the biggest part of the cell cycle, G1, Gap1, copying of the DNA in S, and final preparation in G2. Then we go into the mitotic phase or the division phase, where we divide the nucleus in mitosis and then the entire cell in cytokinesis. So DNA replication happens prior to division, happens in the interphase part. Double-stranded DNA helices will unwind and unzip. A replication fork will form where they separate, and then a replication bubble can be observed where there is active replication going on. Each strand serves as a template for the new strand, so we end up with a semi-conservative replication. And we also have to get this thing started using a primer that starts with RNA, and that RNA will have to later be replaced by DNA. DNA polymerase is the enzyme that attaches the new nucleotides based on the nucleotides that are present in the old strand. It synthesizes both strands at the same time, but both strands are not going in the same direction. One is a leading strand, it can go right along. One is a lagging strand, it takes a little longer, it has to work backwards. There's another enzyme called DNA ligase that will splice the short segments together, particularly in the discontinuous lagging strand. End result, though, is that two identical DNA molecules are formed from an original DNA. During mitotic cell division, one complete copy will be given to the new cell and one will remain in the original cell. So again, this process is referred to as a semi-conservative replication, because each new strand of DNA is going to consist of one old strand and one brand new synthesized strand. So here is the replication fork, and this area back here would be the replication bubble. DNA polymerase is adding strands. It can go right along with the leading strand. The lagging strand, it has to little bit go, reattach and work its way back, and then DNA ligase will tie them together.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, A with T and C with G, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
So that sets us up for mitosis, which is part of cell division along with cytokinesis. There are some cells that do not replicate. Those would include cardiac muscle cells, nerve cells. Skeletal muscle cells don't replace it very efficiently. And in those tissues, we generally get scar tissue replacing the actual damaged cells. The mitotic phase is which division occurs, includes mitosis and cytokinesis, and control of cell division is very critical. If we get uncontrolled growth, we get cancers. So again, here's our total cell cycle. We'll start with M phase, the mitosis, the division of the nucleus. The duplicated DNA, remember that was already duplicated during interphase, is divided into 4 stages for understanding prophase, metaphase, anaphase, and telophase. Prophase is often divided into 2 different parts, early prophase and late prophase. There's a lot of things that need to go on. Prophase is all about preparing for dividing the nucleus. So chromatin condenses, giving us chromosomes. Those chromosomes are two sister chromatids held together by a centromere. The centrosome and its duplicate begin synthesizing microtubules that push each centrosome in opposite poles. It's called the mitotic spindle. There are also other microtubules that forming a centrosome. Later on, the nuclear envelope is going to break up. Special microtubules attached to the centromeres at locations called kinetochores, which are going to pull the chromosomes first to the center of the cell and then away from each other. The non-kinetochore microtubules push against each other, causing the poles of the cell to move further and further apart. So you can recognize a prophase cell by the presence of chromosomes in what used to be the nucleus. Here's late prophase, very clear chromosomes. Nuclear membrane is beginning to break down. And from there we go to metaphase. In metaphase, the centromeres are precisely aligned at the cell's equator. They are on an imaginary plane we like to call the metaphase plate. It's very clear when you use an artist drawing. A little more fuzzy when you look at a live cell because it's the centromeres that are lined up in the middle. The arms of the chromosomes tend to flop around on each side. At anaphase, which is the shortest of all phases, the centromeres split, and each sister chromatid is now separated from the other. The chromosomes are pulled toward their respective poles by motor proteins on the kinetochores, and the kinetochore microtubules the non-kinetochore continue to force the poles further apart. So we're pushing the ends of the cell away from each other and pulling the chromosomes, sister chromatids, away from each other. In telophase, when chromosome movement stops, we start to undo everything we did in prophase. The nuclear membrane begins to reform. The chromosomes go back to chromatin by uncoiling. Nucleoli are going to reappear, and we're going to lose the spindle apparatus. Cytokinesis starts late in anaphase and continues throughout mitosis. Starts with a ring structure of actin called a cleavage furrow, and the two pieces are then pinched apart.
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form 2 separate daughter cells. The cytoplasm of a cell in late interphase contains 2 centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. 1 or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of 2 identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the 2 centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, The spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant...
Since we don't deal with plant cells in human anatomy and physiology, you don't need to know about the cell plate. So there are lots of controls that keep this working the way it's supposed to. There are accelerators or go signals, and there are stop signs or stop signals that direct when the cells should or should not divide. So go signals would include surface to volume ratio of the cell, when the cell membrane becomes inadequate for exchange. There are chemicals, including growth factors and hormones, that may tell cells it's time to divide. Stop signals would include the availability of space. Normal cells exhibit what is known as contact inhibition. When they come in contact with other cells, they stop dividing. Cancer cells do not. There are two groups of proteins that are crucial to go through the S phase and mitosis correctly. One of the best well-studied are the cyclins and the cyclin-dependent kinases. Cyclins are regulatory proteins that accumulate during interphase. Cyclin-dependent kinases are enzymes that put phosphates on other things and activate those cyclins when they bind to them. That cyclin-dependent kinase complex activates enzymes that prepare the cell for division, and then the cyclins are destroyed after mitotic division, and the process begins again. So the cyclin-dependent kinases are always there. The cyclins are only manufactured when they're needed. There are a lot of checkpoints, several checkpoints. There were a couple on our diagram. There are probably many others. There's a G1 checkpoint. That's the most important. If it doesn't go through G1, it stays in G0 and no further division occurs. Again, that's cardiac cells, nerve cells, skeletal muscle cells to a certain extent. Here's that cell cycle one more time. And that's going to do it for this section.